Hi everyone, thank you so much for coming to my poster. My name is Ron, and today I'm going to present the project that I worked on for my honors thesis, which is about the behavior differences in response to opiates among inbred rat strains. So the opiate crisis has long been a serious public health issue in the US, and there's been persistent efforts in research that promotes effective prevention and treatment on opiate use disorder. Genetic variation is one of the most important factors that may contribute to our individual differences in susceptibility to OUD, because having a better understanding of it can provide huge clinical implications. There's now a growing emphasis on studying the biological basis of opiate dependence. However, there's still a lack of knowledge on how opiate use behaviors can be impacted by the specific regulatory mechanisms of genes, such as transcriptional factors. And we believe that it would be an interesting question to explore. One way we can study OUD-related regulatory mechanisms is to thoroughly compare the genetic components between two inbred rat strains that show different behaviors in response to opiates, even when their drug intake is being controlled for. But to conduct such genetic analysis as suggested, we must first identify the two rat strains that fit this description, which is exactly the main goal of our study. In our study, rats from 4 different inbred rat strains were trained to self-administer and eventually made dependent on oxycodone, which is a very common opiate medication. We took their behavior measures on several aspects that were opiate use related, and overall, we expected to see 2 strains that had similar metabolism and escalation of drug intake, but significantly differ in these behavior responses to opiates. And here below is what the data tell us. In the first graph, we can see that the oxycodone metabolism was pretty similar across all rat strains except for ACI. This suggested that ACI had a very different capacity for opiates from the other 3 strains, which were probably resulting in an unwanted difference in their drug intake. However, when we look at their actual intake during self-administration, we found that not just ACI, Qiao also has significantly less opiate intake compared to F344 and BN. So basically, F344 and BN are the only pair of rat strains that had both similar metabolism and similar amounts of intake. And if we look further into the behavior measures specifically of these 2 rat strains, we can actually see that after they develop dependence on oxycodone, F344 displays significantly higher motivation of drug seeking than BN, indicating a difference in their responses to opiates. But what's surprising is that this pattern reversed in the reinstatement session, where it was BN that showed higher levels of seeking behaviors than F344 after 28 days of abstinence. There's also a notable difference between these 2 strains in their withdrawal-induced hypergesia, where there was a significantly larger decrease of pain threshold in F344 than BN during their opiate withdrawal. But there's no distinct difference in their tolerance to the analgesic effects of opiates. And finally, we found that opiate dependence also led to a dramatic reduction of oxygen saturation in F344 compared to the baseline, but it had very little impact on BN, suggesting that BN may be inherently more resistant to the respiratory side effects of opiates. In conclusion, only BN and F344 had both similar metabolism and similar amount of opiate intake during self-administration. And yeah, they show significantly different responses to opiates on several behavior aspects. Therefore, we decided that BN and F344 strains are the 2 most suitable candidates for further genetic analysis. And that's it for my poster presentation. Thanks for the support from my wonderful advisor and colleagues, and thank you all for listening.